what I'd like to kind of ask about is TEDdy. Could you? Because so TEDdy is this very large experiment that you're running looking at type 1 diabetes. And I must admit, I thought type 1 diabetes was like genetic. It was like 100% genetic. But reading your website, I see that it's not. So can you explain, the background to the study and kind of what you found?
Yeah, thank you. And TEDdy is the environmental determinants of diabetes in the young. It's an international consortium that was formed now almost 20 years ago by the National Institutes of Health. And the goal of TEDdy was to identify the environmental promoters or triggers for type 1 diabetes in genetically susceptible children. And as you indicated, there is a strong genetic contribution to type 1 diabetes, but it's not a pure genetic disease. There are very few pure genetic diseases. There are some. And the genetic contribution to type 1 diabetes is probably about 50%, maybe a little higher. But there isn't too much we can do about our genetics. We don't really choose our parents. We don't really control the random events that combine our chromosomes from our parents. So, but we do have control over the environmental factors. And I think that's an important point to make. And we have lifestyle choice, and we have a lot of things that we can control to do something about our genes. So TEDdy decided to look at a large number of newborn babies. And at the end, we screened almost 500,000 newborns by genetic markers. And that was done in 6 clinical centers, 3 in the US and 3 in Europe. And by looking at the type 1 diabetes genes that were known at the time, we were able to identify and enroll almost 9,000 high-risk babies for developing type 1 diabetes. And we embarked on a 15-year monitoring study, and plus 5 years of screening. So it's a 20-year long study. And we are near the end of the study. It's been going on for about 17 years. And we have 3, maybe 4 years to go. And so far, we actually have found a lot of interesting things. And we are also redefining many of the biologies related to type 1 diabetes and also outside of type 1 diabetes as well, such as the microbiome and the natural evolution of our immune system. And it's a very intense and a very it covers a lot of ground, if you want. So we had quarterly blood samples, we had monthly stool samples, and we analyzed the genes, we analyzed the expression of the genes, and we analyzed the connection for proteins, metabolites. We look at the microbiome in the stool and in the blood. And at the end of the day, we wanted to combine all the data together with the clinical data on whether someone has progressed to type 1 diabetes or not, and when the person or the child progressed to type 1 diabetes, And the hope is by looking at all this data together in a large number of children, we can come up with a number of triggers or protective effects for the disease. So we can use this knowledge to design primary prevention for the disease. And we have over I think almost 200 papers published now, and there's a vast body of knowledge and on type 1 diabetes and other biologies. And the key findings are, I think what's really important is type 1 diabetes used to be thought as a very homogeneous disease. And that turns out to be not true. There are many subtypes, and obviously you can define them by various biomarkers or clinical parameters. And being able to clearly define subtypes of disease is extremely important, not only for type 1 diabetes, but also for almost all other diseases. If you cannot clearly define the heterogeneity of the disease, it's very hard to understand what are the causes of the disease or how you can treat. So in TEDdy, we were able to identify several major types of disease, depending on when the antibodies and what antibodies appear, what genetic factors the patients have, and hopefully, I certainly believe the environmental triggers can be very heterogeneous as well. So you can have very different types or combinations of environmental triggers for the disease. And that's why it's so difficult to narrow it down to a single trigger. I don't think there's going to be a single trigger. There are going to be multiple triggers for the disease. We really, and these triggers can trigger the disease in individuals with certain genetic makeup. Again, you have the gene, have the environment interaction, and we really need to understand that so we can come up with the best prevention strategies for the disease. And we're at the beginning of putting the pieces of the puzzle together now, and I think I'm very excited for the next 3 years or so. That's when we are going to figure out what's going on and what we can do about it. And I think it's going to be very, very exciting.
Right. Yes, that does sound really exciting. I mean, to be able to, I guess, offer advice to people who have this genetic risk. Okay, this is how you should bring up your child. That would be, right. Really, really good. I hope that you found the video informative. Professor She is very generous to offer a 10% discount code to our audience for all the tests. Please find the discount code and details in the description. Thank you so much for your kind support. I wish you all well and will speak to you again soon.